Okay, are we? Oh, we're only 10 minutes late. Great. All right, so what we're going to talk about today is we're going to talk about chromosomes. So what I talked about last week was I gave you a little history lesson. I basically talked about all these precursors to modern genetics. And we're just asking, okay, we need to understand the mechanism. We want to know how things work. It's more than just being able to do selective breeding and hybridization. We want to understand what's going on so we can do it better. That's our goal with early days of genetics. Now, of course, the goal is to understand everything even more. All right, so I talked a little bit about Aristotle's version of genetics, which is kind of the dominant one for many, many centuries. And then this young upstart, Charles Darwin, comes along. And he proposes this idea of pangenesis, that there are these things called gemmules that go from everywhere and congregate in the gonads. And those gemmules carry the information to make the next generation. So that's his idea. It involves blending inheritance. It's kind of a Lamarckian model of how genetics work. And it's not going to hold up very well. Lots of people are very unhappy with this particular scheme. What's going to happen next is that, well, what I would say is actually going to happen next is Gregor Mendel is going to come along and propose a better model, but Mendel did his work in the 1860s and then just kind of died, just kind of vanished. It got published and hardly anybody read it. So it'd be nice to say, okay, after Darwin, we get Mendel who explains everything. We don't. What we get instead is in the last half of the 19th century, in the early 20th century, people are doing lots and lots of microscopy. And they're looking at cells. You could argue maybe they're looking for gemmules. Come on. There we go. They're not finding them, but they're finding these other interesting phenomena. So you've taken cell biology. You remember we talked about all those components in the cell and what's going on, mitochondria, ribosomes, all that kind of stuff. They start looking at the nucleus. And the nucleus undergoes some really interesting changes. So what's going on here? Well, when you look at just a cell at rest, its nucleus has kind of a granular appearance. You can highlight that granularity with various stains and things like that, but it's just sitting there quietly. It's not really quiet, it's a really busy place, but it looks like not much is going on in there. But then when the cell is about to divide, radical changes take place in that nucleus. So when I worked on zebrafish, it was a wonderfully dramatic thing to see. You'd put a zebrafish egg on a microscope, and you'd sit there watching, and it would be doing all these divisions. And in between divisions, you'd see these nice round nuclei, and then just suddenly they'd all disappear. Nuclei were gone. And then the cell would divide, and then the nuclei would reappear. So there's something funky going on. Something's happening inside that nucleus just before it divides. This is Walter Sutton. I mentioned him last time. He's that Kansas farm boy. He was doing all these experiments on grasshoppers, a lot like the ones we have around here. And he was also getting distracted all the time working on farm machinery. He's actually going to make a fair bit of money from patents and things like that. But his first love is biology. Let's go figure out what's going on inside that nucleus. And he was taking advantage of microscopy, and he was also taking advantage of the availability of stains, things that selectively stain these things called chromosomes. Chromosome just means colored body. So there's little colored blobs, when you stain for them, that appear in the nucleus when it's just about to divide. Now what you see here is some examples of what he was seeing in the microscope with his grasshoppers. And he noticed a couple of things. These are kind of crucial. He notices that, if you go from grasshopper, from tissue to tissue, and you do this kind of thing, you see patterns. That they've got roughly the same number of blobs. So yeah, you can sit here and count all these blobs. And then you go to a different animal, count all the blobs. They're the same every time. He also could tell, this is the early stages of all this, so he's got a fairly low-level view of what's going on. But he could tell, for instance, there are some blobs that are very small and stubby, and there are other blobs that are nice and long, and there's ones that are in between. And he could categorize those. So he's getting a better idea of what's going on inside that nucleus. That there are these structures, these chromosomes, these colored bodies that are found in the nucleus just before division, that they have a specific number and organization for the species he's looking at, and it's different for other species. This is really suggestive, right? There's got to be something interesting going on here. So he's studying these things. Back in the day, I used to do some of this stuff too. When I was a young graduate student, we did a lot of work with looking at salamander chromosomes. And it's a fairly simple technique that's kind of difficult to master. What you see here on the left, that's a chromosome squash. So, for instance, you take your salamander tissue or your grasshopper tissue or your human tissue, and you put it on a slide, and then you take a cover slip and you put it over it, and you just gently push down until the nucleus pops. Sounds crude, doesn't it? You're just kind of physically rupturing the cell and the nuclei to make the chromosomes sort of dribble out and escape. And you get something like this. Just this hodgepodge of little colored bodies on your slide. Then what you do is you have to scrutinize these and measure them. So this is a terrible example. That's a really hard one to sort out, isn't it? But you can see, some of them are long. So you might pull out a ruler and measure them, figure out exactly how long they are. Some of them are short. There are all these other properties that are present in these, like where is the centromere? And again, you remember from cell biology, you get a chromosome, it's got a little constriction in the middle. That's where the centromere is located. And what you typically do is cut them out. Well, not anymore. Back in the old days, that's what we do. We'd take a picture like this, and we would literally get out a pair of scissors and snip out each chromosome. And then we'd push them around on the table and glue them down, and in order from longest to shortest. And that's what you see up there, top right. So that's a karyogram. It's made from photographs of chromosomes, just laid out, so you can compare them all. So like there's chromosome 1 up there at the top, there's 2 of them. There's our solstice notifications, I would forget that. But you can see that each of these, you have a different chromosome, and all the chromosomes come in pairs. This is typical for a diploid organism like us or a salamander. That there are 2 copies of chromosome 1 up there, and 2 of chromosome 2. What you also see here, this is an ideogram. An ideogram is a schematic illustration of the same thing. Okay, this just reveals a couple of other features. In this case, they're typically all lined up by where the centromere is, and you also see the banding pattern. So if you look at a chromosome carefully under the microscope, you can see there are little regions where it's dark and light that kind of alternate. So these bands are also indicative. When you look at your grasshopper or your salamander or whatever, you stare at it for a long time, you start making these ideograms, you start noticing that every single salamander has a chromosome 1 with a similar sort of banding pattern. Isn't that awesome? So you're starting to get a picture of distinction and differences within the chromosomes of the cells of a specific animal. And you can compare that to all the other salamanders. You can compare it to people. You can compare it to mice. You can compare it to grasshoppers. But it's very suggestive, don't you think? There's something going on here. What is it? Now, Walter Sutton, what he's famous for is his chromosome theory of heredity. What he suggested was, hey, we got these chromosomes that are sorted out in a very specific way and passed on to their progeny. And he also integrated that with Mendel's ideas. This was in the early 20th century when they had rediscovered Mendel. They brought that together and they said, hey, maybe these unit factors, these things that are passed on that Mendel described, are these things, chromosomes. As near as we can tell, he's right. There are some other extra-nuclear things that get passed on. We'll talk about those later. But yeah, he's kind of cracked a secret here. If you want to figure out how things get inherited, look to the chromosomes, is what he's saying. Okay. Advance. So here's a list of a couple of specific principles that we found with chromosome arrangements. So organisms have a specific base number of chromosomes. It's called N. When we talk about the N of an animal, we're talking about the number of chromosomes it has in its haploid cells. So we'll talk about that in just a second. So in humans, for instance, we have 23 pairs of chromosomes in all of our cells for a total of 46 chromosomes. That's what's listed over here. If we looked at a fruit fly, Drosophila has an N of 4. So it has a total of 8 chromosomes. Don't get confused when I say N is 4 and then I point to this table that says 8. That's because fruit flies and people as adults are diploids. That means we have 2 copies of each of our chromosomes. I showed you that in the previous slide, right? So you got 2 copies of number 1, 2 copies of number 2. That is something called ploidy. Okay, what does ploidy mean? It just means that is the multiple you use to multiply the number of chromosomes in N to get the actual number of chromosomes you see in the cell. So you adults, we're all diploid. So we have a ploidy of 2, and we've got 23 chromosomes. We have an N of 23. So that means our total in each of our cells is 46. See, this is just really simple math, isn't it? We don't get into the complicated math in this course. Okay, but what's relevant too is that sperm and egg are going to be haploid. So they're just going to have 1 copy of each chromosome. And that means that in sperm and eggs from anyone in this classroom, if we looked, we're not going to, okay? Just clear that from your mind. We're not going to look. But if we did look, we'd see that sperm and egg each have 23 chromosomes. So their ploidy is 1. Later in this course, we're going to talk about some other examples. Things like triploid, tetraploid, octuploid, you name it, any multiple. So we're going to have different ploidies in some organisms. Now, the important thing to remember here is that in diploid organisms, chromosomes are always found in homologous pairs. Again, with some exceptions. Isn't this great? I'm going to be giving you these general rules for the first couple of weeks of the class, and then we're going to spend the next part of the semester telling you all the exceptions where this rule breaks down. But just in general, you can say, okay, chromosomes are found in homologous pairs in diploid organisms. Okay, we also can recognize these chromosomes, as I mentioned previously. We can pull out the chromosomes, we can measure their length, we can measure the position of their centromeres, we can look at their banding patterns, and we can identify homology. So I have 2 copies of chromosome 1, right? And I could take a blood sample from each of you, and we could do a squash, and we could look, and we'd see that all of you also have 2 copies of chromosome 1. And on a coarse level, just looking at the morphology of the chromosome, we could probably say that our chromosome 1s look very, very similar to each other. If you get down into the details and you look at the sequence of nucleotides, then we start seeing more differences. But in general, we're going to see that there's a pattern that's maintained. Okay, and as you can see from my little table up there, there's a huge amount of variation in chromosome numbers. N varies all over the place. N also doesn't seem to say much. Yeah, it doesn't say much about the organism at all. It's the content of the chromosomes that matter. The way I usually describe this is, think of chromosomes as big filing cabinets. Each filing cabinet can hold thousands of genes. And it's just organizational. Yeah, we got the same genes. Humans and chimpanzees, for instance, have almost the same genes, but there's a couple cases where they move them into different filing cabinets. But we can still find them there. Mice, where's mouse? There's mouse. Mouse has 40 chromosomes, an N of 20. And if we compare mouse chromosomes to human chromosomes, we can find the same chunks of data, just broken up in a different way in different filing cabinets. That's a property called syntennium. We'll talk about that later in this course too. Okay. So I mentioned ploidy. There's a couple of specialized cases of ploidy that I'll mention here, 2 broad categories. One is euploidy. That's mainly we're going to talk about it first. Euploidy says the number of chromosomes is an integral multiple of the haploid number. What does that mean? That means that, for instance, gametes, which have half the diploid number, so human gametes have 23 chromosomes, adult humans have 46. Both of those are euploid. They're euploid because you just got to multiply by an integer to get the right number. Similarly, an individual who is trisomic, or who is triploid, there's no such thing as an adult living triploid human being, though. It's lethal for all humans. But if there were a fully triploid individual, who would have how many chromosomes? If haploid is 23 and diploid is 46, what would triploid be? You can do the math.
69.
Yes. So 3 times 23. What would a tetraploid be?
92. 82.
No, 92 is the right answer first. Yes. Yeah, so this is really easy math, right? Okay, there are also special cases though. This is called aneuploidy, where an individual has lost or gained a single chromosome, sometimes a couple of chromosomes. Aneuploidy means you can no longer get their number of chromosomes just by multiplying the haploid number by the ploidy. Probably an example you're all familiar with is Down syndrome. Down syndrome is a situation where the individual has 3 copies of chromosome 21 and 2 copies of all the others, and the total number of chromosomes they have is 47. 47 is not an integral multiple of 23. Okay. So we will later in this course talk a little bit more about aneuploidies. These have severe consequences in people. You can have cases where an individual is monosomic and they're missing a whole chromosome. So you can have an individual who's got 45 chromosomes, or somebody who's trisomic for one, and then they would have 47. And there's all kinds of variations on this. But again, we're going to set that aside for now. When we get to talking about all the exceptions, then we'll discuss those in more detail. Okay, how do you make a polyploid? That is somebody that's not diploid. How do you make a triploid or a tetraploid? And it turns out that happens fairly easily. It's a common consequence of a couple of things. You notice the distinction here made. We've got autopolyploids. An I got to pronounce these names. Autopolyploids, what are they? Well, they are individuals who have had a failure of the chromosomes to separate in meiosis. All right, so all of you are sitting here with 46 chromosomes in each of your cells. And it's also true in your gonads. But then to produce sperm and egg, what the cell is going to do is go through a very specific kind of division to produce a gamete with 23 chromosomes total. And then, of course, fertilization is you take 2 cells with a chromosome number of 23, mix them together to get the 46 back. What happens if you're supposed to make a sperm cell, for instance, with 23 chromosomes, and something disrupts the nuclear division? You can make sperm cells that have 46 chromosomes, or egg cells that have 46 chromosomes, just by suppressing a division. And as you see here, there's lots of ways you can do that. Heat shock, cold shock, they both work. There's also things like colchicine you can treat cells with. And what all those do is they disrupt the cytoskeleton of the cell so it doesn't carry out a proper division. So you basically shut down its ability to share out, split up the chromosomes, and you end up with a gamete with, for instance, 46 chromosomes. And then that gamete, for instance, could be then go out and fertilize another cell that has the appropriate number 23. And then you end up with an individual that's got 69 chromosomes. And then they die in utero, so you're not going to see them. But in some organisms, we can do this. Okay, so that's one case, is autopolyploidy. Just involves one organism, one species, one mating event, for instance. Allopolyploidy, allo just means different. It's the result of hybridization between 2 closely related species. So we're going to combine 2 species. This is really common in plants. We do it all the time in agriculture. Not so common in animals. Okay. Here's just to illustrate this phenomenon. So we've got an organism. What's the N of this organism? What did you say? It's 2N. Yeah, it's a diploid. But what is that? So its ploidy is 2. But what's its N? What's the haploid number of chromosomes? 3, yes. Okay, so this one is a diploid organism with an N of 3, so it's got 6 chromosomes. And what could happen is you suppress one of the divisions, and you can then fertilize with another cell, and you can end up with triploid or tetraploid organisms. This is just autopolyploidy because it's not involving any other species. So we're just breeding these organisms together, and we've suppressed one of the nuclear divisions, and we get these anomalous results. As I said, we can do this in plants. It happens fairly routinely in plants. I believe I mentioned before that strawberries are octoploid. How do we get octoploid strawberry? Well, this is one way. Just a failure of division in the gametes. Allopolyploidy is a little messier. So over here, okay, we got 2 diploid organisms. What's the N of this individual? 2, yes. This one is 2 as well. So we'd say, okay, N is 2, really simple. We can figure that out. But then, through some freak of nature, these are 2 different species. Look, we've drawn the chromosomes in different colors. The plants themselves may look different from each other. But somehow, through a freak of nature, we get pollen fertilizing the ovule of another one. And we get this situation. What's the N of that cell? Yeah, in this case we'd say it's 4, because these chromosomes are distinct from these chromosomes. So there are 4 different kinds of chromosomes. The ploidy is 2, so that's what we get. Now, this can get, we can make this even messier. So that example, we had one plant with an N of 2 and another plant with an N of 2. What you see illustrated here is, imagine we get 2 species. And the first one, this is a diploid here at the top. That first one has an N of what? Count the pairs of chromosomes. That one's 3, yes. And we're going to do this terrible thing of breeding it with a completely different species up there at the top right. That one has an N of 2. So they are going to produce gametes, haploid gametes. These are perfectly normal, healthy gametes from these 2 species. And then we're going to force them to come together. And we get the result right there. The AB cross. What's the end of that one? It's going to be 5, sort of, but they're not in pairs, are they? We don't have homologous pairs anymore. Yeah, there's only one B1 chromosome, one B2 chromosome, one A1 chromosome. So we don't have homologous pairs. This is a diploid. You're supposed to have homologous pairs. And we don't. What will happen? Well, the plant itself might be perfectly healthy. It'll grow. Maybe you'll do this and you just go, oh, this is making really tasty fruit or something. Or big, large kernels of corn or whatever we care about. But it's sterile. Why is it sterile? Because when it goes into meiosis and it tries to create new gametes, It can't. There's no easy way to sort these out. I mean, you could imagine, okay, well, we're going to go ahead and make gametes, and okay, one will have this, these three, and the other will have this too. No, that's not viable. That's a messed up organism. So one of the ways we can rescue that, though, is we then make it an aneuploidy. We double all of its chromosomes. Yeah, so we're just going to induce polyploidy. We're going to suppress cell division. And what do you get? You get this organism here, which now has A1, A2, A3, B1, and B2. This combination that seems to work very well. But now each of them is in a pair. And if they're in pairs, then we can go through meiosis, and we can produce new gametes. So we're going to produce new plants from this. The new gametes will look like these two. I know it sounds like magic. And maybe you're struggling to wrap your head around all this. But yeah, that's one of the obvious things you can see just looking at the levels, at the level of the chromosomes, is that you can do this sort of thing. There's another common phenomenon that goes on. And this is called Endopolyploidy. So it's kind of an internal polyploidy. You guys, you've got polyploid cells all over in your body. And not in your gametes. Gametes that are polyploid, that would interfere with their function. You wouldn't be able to have progeny. But you can take somatic cells and make them polyploid, and that has some advantages. So these are cells that have more than the diploid number of chromosomes, and some examples are Drosophila salivary glands. Drosophila salivary glands are great for studying chromosomes. Why? Because the larvae have these gigantic salivary glands. They're huge. They fill up much of the front half of the animal. And that's because the way the Drosophila larvae feeds is it goes burrowing through the medium, you know, that blue stuff you got up in the lab, that you'll have up in the lab this week. It goes burrowing through the medium, and it just pukes up huge amounts of saliva that then digests the medium, and then it slurps it back up. So you got to make lots of saliva if you're a fruit fly larva. Has anybody seen the movie The Fly with Jeff Goldblum? No? Classic horror movie. Guy turns himself into half human, half fly, and then he goes to show off to his girlfriend how he eats now. He brings out some donuts, and he just pukes up all this saliva on them and sucks it back in. That's what your flies are doing. That's how they live. Okay, you say, you are not a Drosophila fruit larva. You do not eat that way. The other place we find these is in, for instance, the liver and your brain. Liver is an extremely active organ metabolically, as you all learned in cell biology last semester, right? Doing all that processing of glucose and sugars and storing glycogen and cool stuff like that. It's a very busy organ. It needs lots of enzymes to do its job. And so vertebrate liver has cells that are tetraploid or octoploid. They just multiply their chromosomes many times over because then they can make more enzymes rapidly. Here's another one. Yeah, if larvae of fruit flies live by producing huge amounts of saliva to digest their foods, we human beings make big brains, right? We got gigantic brains that are doing all this fantastic stuff that are keeping you awake while I'm lecturing at you. And many of these neurons are also polyploid. Pyramidal neurons in the cortex, for instance, these large neurons that send axons for long distances, and they have a huge cell volume, tend to be polyploid. So we're going to have lots of that going on. It's just so they can produce lots of the byproducts that you need in order to carry out metabolism in these cells. So as you see here, endopolyploidy is often associated with cells that may be unusually large or that are involved in extremely active metabolism. However, I don't need to talk about these anymore because they're not really involved in genetics. Yeah, you don't breed by sharing brain cells, right? Or saliva, whatever. So these are specialized cells, specialized somatic cells that have very specific jobs to do. And they're kind of outside the domain of reproduction. Point being, just that polyploidy is a common solution to lots of situations. Okay, what do we got here? This is a little diagram of what a chromosome is like when you look really closely at it. So, when you take apart a chromosome in the microscope and look very closely, there's a couple of features that should jump out at you. We've already mentioned one, centromeres. Centromeres are kind of an anchor point for fibers to attach to pull the chromosome around in the cell. So this is the handle for moving chromosomes around within the cell. I've also mentioned these bands, these dark and light bands. So you find these dark bands are made up of heterochromatin. That is DNA plus specific proteins that wrap around them. And usually, typically, heterochromatin is inactive. It's regions of the chromosome that have genes you don't need or that are full of junk DNA. So you just kind of shut them down by wrapping around with these proteins. Then there's euchromatin. Euchromatin is the light areas. And these are typically associated with more active transcription going on. Not always, though. But anyway, there's complications there. Okay, so we've already mentioned those. The other thing is, you can look at the shape of the cell, or the chromosome, and you can see that there's often a short arm. That's the petite arm. It was named by French investigators. Okay, so they called this petite. I think that's really cute. So it's just the short arm. And then they called the long arm the Q arm because Q comes after P, I guess. I don't think there's a French word for long arm that starts with a Q. Okay, so we got P arm and Q arm. One of the things you can do with this really rough piece of information is you can use this to categorize the chromosomes. So that's what you see over there on the left. Is we have a nomenclature that we can use to define the position of the centromere in the chromosome. So there's a telocentric chromosome. So you can see that the centromere is way up there at one end. So the petite arm is, if there is one, it's pretty much negligible. And then there's acrocentric. We got a really short P arm and a really long Q arm. Then we got submetacentric. Submetacentric just means the centromere is just off of center. And as you might guess, metacentric means that it's right in the middle. There is no difference in length between the P and the Q arms. They're the same length. As I mentioned way back in the distant days when I was doing this stuff, this is one of the parameters we'd measure in these chromosomes and salamanders, is how long are these two arms? And then we take a ratio called the centromeric ratio. So we assign a number. So metacentric, obviously, it has a number of 0.5. It's halfway down. And heliocentric, well, it's basically a 0. So you can do all these measurements get a number, and you do a bunch of your salamanders, and you can then group them not only by their length, but on their centromeric ratio. Okay, all that's really useful information if you're doing a taxonomy of chromosomes. Some of the characteristics that are important to remember about chromosomes are listed there. So you can tell chromosomes apart. That was a revelation. So we can actually categorize chromosomes by their size, their centromere location, and by that banding pattern. We also see something shown here, that at the start of cell division, when you look, each chromosome consists of 2 strands of DNA. That's what we're seeing here. There's one strand, here's another strand, they're linked at the centromere. Each of these is called a chromatid. Okay. This is one chromosome, but it's got 2 chromatids. One of the tricks you'll use when you're looking at a cell and counting chromosomes, you don't actually count the arms, you count the centromere. So asking how many chromosomes are in that cell is the equivalent to saying, okay, how many centromeres can you find? As you see here, we got 2 chromatids. These are also called sister chromatids. Why are they called sister chromatids? Because they're identical to each other. So they're identical twin sisters. This arm has the same nucleotide sequence as this arm. And that's also going to be important later when we start talking about recombination. In addition, each cell, each chromosome, has a homologous chromosome. And I know it's sometimes easy to get those confused. So this is 2 chromatids. If this were, for instance, chromosome number 1 in our organism, there would also be another chromosome we could draw over here with the same arrangement of bands, the same size of the arms, the same relative location of the centromeres. Okay, that's different from sister chromatids. And we call those homologous chromosomes, and they are not identical twins. They're more like fraternal twins. They've got basically the same stuff in the sense of the same genes for particular organs, but they got different forms of those genes, which we call alleles. Okay, so for example, you could imagine that on this chromosome, there's a gene in here for blue eyes. And we got chromosome 1 duplicated over here. It's also got a gene for eye color, but it might be like for brown eyes in there. So keep those straight. Sister chromatids and homologous chromatids, or homologous chromosomes. They're different concepts. Uh-oh. Come back. There we go. Now, as I said, way back in the dim, distant days, the Middle Ages when I was doing this stuff, we would do it the crude way. Scissors, cut out the chromosomes to make our karyograms. Or when we got really fancy, we got little digitizers and we'd measure out with a digitizer along the length of each chromosome and we get these nice numbers. Obviously, nowadays, we are much smarter than that, so we do it this way. This is a technique called fluorescent in situ hybridization. It uses molecular probes, colored probes, that bind to specific regions on specific chromosomes. Basically, what you can do is you can just throw this on your chromosome squash, and it's not just every chromosome comes out the same color. Now it colors the chromosomes for you. So you get these nice blue ones, and pink ones, and yellow ones, and blue ones. That's a great step forward. It makes it really easy to tell them apart. And the other thing that's really gratifying about it is you can nowadays just aim your computer at it, get a camera, and you point that at a photograph of your squash, and the computer will sit there and it will stitch out all the different chromosomes and splice them together for you in a nice little karyogram. And sometimes it'll even take that karyogram and turn it into an ideogram. Yeah, that would have saved me so much work in the 1980s. So in some ways, progress is wonderful. Okay, so we can do all this stuff looking at these chromosomes. Oh, I got 5 minutes. Great. I'm not even going to get to mitosis today. Wednesday, we'll get Wednesday. We'll hit mitosis and meiosis. Okay, so you remember this from cell biology, right? The cell cycle, G1, S, G2, and then the M phase where mitosis occurs. I've just added a little bit of information relevant to us here. So during G1, all of the chromosomes are single-stranded. So no sister chromatids, okay, just a single strand of DNA, all wrapped up in protein. And they are also dispersed. They're all just kind of spread out. Okay, so everything's accessible to proteins in the nucleus. Then we go into S phase. And again, think back to cell biology. During S phase, there's all this stuff. These polymerases come along, and there's, oh, you remember all those different enzymes, right? We had all these enzymes we went through that are involved in replicating the DNA. And what we get out of that is 2 strands attached together at the centromere. So in G2, we're going to have double strands, and we're also going to be still dispersed, still all spread out. Then we're going to enter into this mitotic phase. We're actually going to divide the cells. And now what's going to happen is we're going to get condensation of those chromosomes. Basically, chromosomes are just going to get supercoiled, just going to get wrapped all around each other. They're also going to get wrapped around with other proteins. We're going to get thicker and denser. So we get something that I've cartooned here like this. So at the start of mitosis, the chromosomes are all double-stranded because they've been through the S phase, and they're also condensed. This is why Walter Sutton would see those colored blobs. During interphase, you wouldn't see those. You just see a boring nucleus. But then at the onset of division, all the chromosomes condense down, they get very dense. You can now stain them with various agents, and you can visualize them as these discrete little blobs. They're then going to go through mitosis, which I don't think we'll get to today. Oh man, I'm falling behind already. It's only the second lecture of it, of course. Anyway, so we're going to see that what you've got to do then is you've got to divide these up. You've got to split those sister chromatids apart. And those sister chromatids are going to move to separate sides of the cell, and then division will occur. So at the end of mitosis, our chromosomes look like that. They're single-stranded. We split them apart, the sister chromatids, and they're condensed down, nice and thick and easily seen, relatively easily seen. And then as we go into G1, what they're going to do is they're going to uncoil and spread out and get back to this state, single-stranded and dispersed. Okay. So, what we will talk about on Wednesday, just to give you a quick preliminary summary, is mitosis. Mitosis is a process that precisely segregates duplicated genetic material so that each daughter nucleus receives a complete copy of the genome. So this is the key about mitosis. When cells divide by this process of mitosis, the 2 daughter cells that result are genetically identical. Okay, we're going to talk about meiosis, which is different. But these cells are going to be genetically identical. The genetic material is split by a process called karyokinesis. Karyo just means nucleus. So we carry out karyokinesis that splits the nucleus. And then usually after that, we also carry out cytokinesis. If you don't carry out cytokinesis, you get endopolyploidy. You just accumulate extra copies of the genome. Then here's what we're going to go through on Wednesday. The 4 stages: prophase, metaphase, anaphase, telophase, and how we take those synapses, those chromosomes that are linked together, those sister chromatids, and we're going to split them apart. And that's how you go through mitosis. Hey, we just did it. I don't have to come in on Wednesday. No, we do have to. There's details. We'll talk about details on Wednesday. Okay, I'll let you go. We'll see you in lab. Remember to review the lab procedure. Yes?